Hello, my name is Dr. Roderick L. Roll, and today I will be talking about active processes. Active processes will require energy in the form of ATP. So, active transport is going to be when we move molecules from an area of lower concentration to an area of higher concentration. This is going to require energy. So previously we talked about moving molecules downhill. Now we're going to talk about moving molecules uphill. To do this, we're going to need a carrier protein. So in this image, you see this carrier protein moving that green square up its concentration gradient. So there's 10 sugar molecules inside the cell and 2 outside. I'm going to take this sugar and push it up its concentration gradient. In order to do that, I'm going to need some energy. This slide illustrates I have a high concentration of sodium outside of the cell. If I take one of those sodium molecules that's inside the cell and push it outside, that would be an active transport. Here is sodium being moved against its concentration gradient. Active transport has many types. The first 2 types we'll look at are called symport versus antiport. Symport system is when 2 substances are moved across the membrane in the same direction. If we move sodium and chloride outside of the cell at the same time, then that would be a symport system. Antiport is when 2 substances are moved across the membrane in opposite directions. Take that same scenario and take sodium and push it out and potassium and push it in. Simultaneously, that will be an antiport system. Let's look at this picture to explain that concept in much more detail. We have sodium binding to the carrier protein. Energy is required. Phosphorylation occurs where the energy is going to be removed from the high energy phosphate bond, and I'm going to release that energy. I'm going to form a DP and a phosphate. A conformational change is going to take place once the energy is used. That conformational change is going to take the 3 sodium that were inside of the cytoplasm and push them outside into the ECF against their concentration gradient. So 3 sodium moved out. Now potassium is going to simultaneously move in. So the potassium binds to their active sites. The potassium is now in the active site. The conformational change occurs, therefore releasing potassium into the ICF. So 3 sodium moved from the ICF to the ECF, while 2 potassium moved from the ECF to the ICF. This occurred simultaneously, so which type of active transport is that? Active transport can also be broken up into primary active transport and secondary active transport. So let's look at primary active transport. The hydrolysis of ATP phosphorylates the transport protein, causing a change to occur, a conformational change. This moves the ions against their concentration gradient. Three examples of primary active transport are calcium, hydrogen pump, and sodium potassium pump. The sodium potassium pump is seen in animal cells, especially in neuronal cells. The hydrogen pump is seen in bacteria, fungi, and plants. And the calcium pump is seen in animals also. In this image, notice that we have 3 sodium ions in the ICF and 10 sodium ions in the ECF. We're going to push some of those 3 sodium that are inside outside. We're going to push it up its concentration gradient or against the concentration gradient, therefore making it even more concentrated in sodium outside. So we'll have 11 sodium versus 2 inside. This is going to create a slightly positive charge on the outside of the membrane versus the inside of the membrane. Why? Because we have so much sodium outside. And these sodium ions are cations, meaning they have a positive charge. So if I have way more positive charged ions outside of the cell than inside of the cell, the membrane is going to have poles. It's going to have a positive side and a negative side. This brings us to the concept of an electrogenic pump. This is going to generate voltage across the membrane. Calcium, hydrogen pump, and sodium potassium pump are all electrogenic pumps. What this means is they're going to generate some kind of electrical charge. In reference to the previous slide where you see the positive and the negative on the membrane, that is created because of these pumps. How is the electrogenic pump relevant? When you take anatomy and physiology, you're going to talk about the action potential. This action potential is how we contract our muscles. The action potential is going to move down our neuron. How this occurs is through the charges associated with the membrane. Remember, because sodium is being pumped out at such a high rate, the outside of the membrane is going to be positively charged versus the inner portion of the membrane is going to be negatively charged. The electrogenic pump generates voltage across the membrane. This is showing you how that action potential moves. The way the action potential moves down the neuron is nothing more than open and closing doors. The doors in question are going to allow sodium to continue to go out or to stop sodium from coming out. If sodium is pushed out against the gradient and the outside is positive, if I close the door, then I can momentarily make the outside not positive. The change in that charge is the action potential. So as you propagate, as you move down the membrane, of this neuron, you just open and close doors, therefore changing the charge of the membrane. The second type of active transport is called secondary active transport. It is going to use an exchange pump, such as the sodium-potassium pump, indirectly while it moves another type of solute. So as sodium comes back into the cell down its concentration gradient, glucose is going to hitchhike and catch a free ride. So remember, energy is used to push sodium outside of the cell. That sodium has to eventually flow back downhill into the ICF. And when it flows into the cell, glucose binds to this protein and comes into the cell with sodium. So glucose will catch a free ride through a co-transporter against its concentration gradient. So glucose is actually going against its concentration gradient while sodium is going down its concentration gradient. This picture illustrates that concept again. To the far left, you have the primary active transport. Sodium is being pumped out. That sodium has to eventually come back downhill. When it flows through the co-transporter, glucose hitchhikes and catches a free ride into the ICF. That is the secondary active transport.